It's my pleasure to introduce Sarah Hart-Johnson. She's the director of the Biological Research Facility at the Francis Crick Institute in the UK. So Sarah has an extensive background in the production, preservation, re-derivation of genetically altered rodents and zebrafish. For 10 years, she has managed the genetically engineered model facility at the Medical Research Council's National Institute for Medical Research, where she led the re-derivation of over 1,800 mouse lines and also helped establish the new facility. In 2019, she set up the colony management service at the Crick. And in 2023, she became the director of the Biological Research Facility, where she continued to drive advancements in the field. So today, Sarah will present her talk on colony management, efficiency, and management at the Crick. Welcome.
Thank you for that introduction. And thank you for inviting me to be here today. It's really great to be part of this type of event. And I've already been scribbling away ideas from that last talk. There were some really fantastic ones, and also from the questions that came up. So I'm hoping to give you a bit of a taste of how the Crick tackles our responsibility towards refinement and reduction with regards to the breeding of genetically altered mice specifically, and some of the strategies that we've put in place that we think are effective in driving down numbers of animals used and surplus. But I think it's fair to say that it's always a work in progress, and it's a journey that we're all on. So obviously, to give you a little bit of background, the Crick's been operational since about 2017, and it was formed from the merger of two large establishments, the MRC's National Institute for Medical Research and Cancer Research UK's London Research Institute, together with funding from the Wellcome and the Imperial, UCL and King's College London were all our funding partners, and we work closely together with them. So that start in 2017 meant that we had staff coming together who had a lot of experience, but we had the opportunity for a new start, and it gave us the chance to look at our lines, look at our policies, and refresh the way that we work. So we have over 100 research groups, and about 60 of those employ animal models for their research, predominantly mice, but also zebrafish, high numbers, and we have various other species. And across the mice, we have 5 main barrier facilities at slightly different health statuses, but more kind of different subject specialities. And currently, we've got about 20,000 cages occupied, but we can take about 27,000 cages. So the key aspects of our strategy that I'm going to talk you through that we think all have an impact on driving down surplus and really improving the refinement and reduction and the refinement of how we use our models and reducing animal numbers are the fact that we have a dedicated colony management team, the fact that we spend a lot of time supporting and training our staff and scientists to increase their expertise in colony management, and we have a real focus on colony planning. We also provide guidelines to optimize breeding efficiency, and those have a focus on genetic integrity and on reducing breeding surplus. And we have a very comprehensive database which was developed at the Sanger, and that gives us very good record keeping, which also means that we can interrogate that data and really find out what's working, what's not. And we have a lot of tools that we've built off the back of that kind of highlight areas of importance and help us to kind of focus on where we can make a difference and drive down surplus. We also try and incentivize and make things easy for our scientists. So we provide core wild type and GA lines to ensure reliable and reproducible supply and to try and reduce duplication, and a free cryopreservation and IVF service to really engage our scientists with our reduction policy and kind of help reduce surplus by taking those lines off the shelf. So I'm going to talk about all of those in a moment. And I think the other thing we do is we take advantage of opportunity. So whether that was when the Crick opened and we were able to interrogate all the strains that were there and question what was needed, where was the duplication, what was the best background. the opportunities in many ways that COVID provided in terms of us having to look at trying to reduce lines. And we went into detail again with every single line on the shelf. And also, you know, in terms of just, I suppose, budget and finances are always an issue. And that gives us another incentive that sometimes the scientists obviously respond to well, that help us to drive down costs, because whilst reducing animal numbers is something that we want from an animal welfare perspective, it also obviously has cost benefits and savings. Oh, and so since the team started operating, we've had about a 30% reduction in the number of live strains that are maintained on the shelf, and we've dramatically dropped the cage occupancy and the number of animals used, and that shows in the statistics that we report annually to the Home Office, which is the UK regulator of animal science. So I think probably one of the biggest impacts on colony management practices has been setting up a dedicated team without direct animal husbandry responsibilities. So again, quite similar to Danielle and Filippo were talking about. And I have a really fantastic team led by Clive da Costa, who is head of colony management services. And we've got 5 staff who are focused on providing support and really simplifying colony management for the sort of 550 or so scientists that we work with daily. And that team provides complete colony management for all of the new groups starting at the Crick, and they do that for sort of 12 to 24 months. And that's been a really effective way of getting those groups and those scientists engaged with the importance of good colony management. So we spend a lot of time teaching them about good practice and sitting with them and working out effective colony plans, which are regularly reviewed with the group. And then after that period, we do hand it back, but that they work well with us. They still come back to us for advice, but they're they're much more experienced and knowledgeable. And that team does a lot of genetic validation, both genotyping and genetic monitoring, and also sometimes microbiome monitoring. Engaging our groups early in the advantages of knowing and maintaining a reliable background and the impact that has on their research. We also, this is the team that kind of does much of the training, trying to improve the expertise across the Crick, which is so critical to effective and efficient management. So training to animal users is provided, and the tools I've talked about. And something else the team do, which is spearheaded by Emma, who's someone on my team who's watching, which is they take on projects and they really look at bottlenecks in the process, and they look at process flow and how we can improve it to drive down on animal numbers, kind of shorten time, the time needed by the scientists to spend doing things that we think could be automated, so that they can focus on the real things like the decisions around colony planning. And I think genotyping is a really good example of that, where we did a lot of digging into when the animals are genotyped, you know, when are the air clips taken, what day of the week, were there any issues in the transport and in the use of the automated genotyping company that we use, which groups were adding that information back onto the system and then actioning the genotype so that we can take cages off before weaning if they are of the wrong genotype. So Emma really worked to streamline that whole process, and a big part of that was working with our database to amend it so that we could automate the translation of genotyping results. So we have a really slick process now where they're all done, or a majority of them are done by day 15 of age. They get sent off, and when the results come back, it's a click of a button and the results will go into our database, and there is a translation in there for every single allele, and the complex ones as well, that then gives the results are in there for each assay. And the thing that we're working on now, and that works with some of the groups, but we've still got a long way to go, is going, right, well, you've got the results of your genotyping, before 21 days, the mice haven't been weaned, please now put on the tasks to action that, so that the cages are off the shelf as soon as possible. So that team have had a real impact, and they work very closely with our genetic monitoring team. Training and increasing staff expertise is the other key way to improve efficiency and knowledge of the ways to drive down surplus and costs. And I strongly believe that by managing our strains more efficiently and characterizing our models properly, we have better models, which makes for better science. So our training covers the basics to advanced colony management. It's really important to start with the basics, such as the mouse's reproductive biology, so that scientists who are completely new to lab animal research know when to set up the animals and how to really maximize productivity. So we train them in a variety of ways to try and reach people who learn through different ways. So whether that's one-to-one or course-based ones. But all of our scientists also have to do a compulsory course in colony management, and then have a short assessment, which will then demonstrate competence, and they have to repeat that every 3 years. We also have an annual refresher for all animal users, where we go over essential changes to legislation and practice, and part of that is an update to colony management and whatever our current drive is to try and improve refinement and refine our work. So I think understanding a strain or a colony is really essential to be able to plan the breeding scheme and to meet the needs of the science and to minimize waste, whilst also ensuring that welfare is maximized. And to that end, being able to communicate in a clear fashion what the strain is and any expected phenotype is critical. And we do that on our database. We have some very clear colony notes that we require people to fill in, so that the technicians looking after the line can clearly see what it is, kind of any phenotype, what the mating schedule should be, and kind of what they're sort of aiming for. And it should also have details of what animals can be culled, what genotypes. So that's something that we're working on very hard at the moment. And anybody who's new in the past few years has this in place, but many of the other groups we're still working on. And we also are trying to get that reviewed kind of every 6 weeks, because the needs of the science change and it grows or shrinks, and we want to make sure that production is in line with demand. So accurately sizing your colony, there's lots of things that you need to know. You need to think about whether it's a kind of breeding feeder line that's going into producing an experimental, or is it an experimental line? What volume of mice do you need? How often? The sex, the genotype, the age. You need to be asking all those questions, and you need to be asking them regularly. And then you also need to think about your strain. What are the peculiarities of the strain, the impact on health and reproduction or Mendelian ratios? Do you need controls? Which ones? And what is useful to you and what is unavoidable surplus? And I think doing this planning and regularly reviewing and adjusting colony size to meet demand is probably the single most important thing that you can do to minimize surplus. So there's a lot of resources out there that can help. So if you don't have a team in-house that can help you, or like we do, there's a lot of things out there to help you. We still go to them regularly. The NC3Rs, the UK's organisation, has a breeding and colony management hub, which has some really great resources, and it works through various scenarios and gives you some possible solutions as to how you should be managing your lines. They also have a really nice worked example of intermittent breeding, which I think there was a question I spotted earlier about if you've got a paper and you're kind of keeping your line on the shelf, waiting for that paper for the review. You don't know what they're going to ask, and it's actually an example that kind of gives you good big cohorts that you let breed, and is one nice way to manage a line. Although there are others that you can use, such as freezing strains. And so the JAX has got great courses, and they have a breeding colony calculator that we use for some of the regular lines that we want to breed for a long time. And then when we're trying to work out experimental cohorts, we find the calculator from Zurich really great that was produced recently. So we also try to provide clear guidelines to help outline best practice. And this is just one simple example. It's very visual that we have around, just to kind of remind people of when they should be setting up their breeders, separating them, and that sort of thing. And it is very effective. We have a really good database that we're very happy with that lets us track all of our animals and clearly task them as well. So we can go in and we can pick the animals that we want to be set up for breeding or for various experiments. And that helps us to track all our animals, but it also helps us to interrogate the data. And that is, I think, incredibly important. So I've popped up a picture of Chris here, who's on my team, who's a real whiz, and together with Tom, who works for him, they have built us a lot of tools that are really there to help us analyze our colonies' productivity, what they're used for, kind of highlight health concerns, and really much more. And the aim of that is to make a tool for our scientists to focus them towards the areas that need attention. And we have a lot there as well that alert us to potential problems across the whole facility, or also groups and labs that might need a little bit of additional help. And this report shows the groups how many lines you can drill into it, and you can start finding out about every singly housed animal. Or you can see this group has a 23% experimental usage of the animals that they breed. And then you can kind of drill into that for the strains and try and work out what needs doing. And the key here is really to make it easy and to focus their attention. So we have a lot of reports, like one that is for matings, and it shows you which matings are starting to be unproductive, that might have high numbers of total litter losses, or they've been unproductive for a period of time. And we set criteria where we flag them with a yellow flag when there's something to keep an eye on, and they go red when we think, well, you really need to be replacing these by now. We have reports that look at productivity. So the productivity index is how many pups are weaned per female per week for each strain. And we can drill into the impact of different mating setups, different breeding schemes. And we've got reports that do really complex multi-allelic crosses as well. And this is really helpful for deciding when we need to change strategy to increase or decrease breedings, when something is very different from its background strain and something might be going on. Where low numbers are needed, we can look at delaying breeder setup or intermittent breeds. So some of these tools just help us to come up with, interrogate what strategy we should use next. So we have a report on singly housed animals so that we can focus them to areas of welfare concern. We look at the strain's mortality rate, and we can do this across the lifespan, but we can also do it via age, and we can highlight different health concerns. And we do that against the background strain. So we're highlighting which strains that you really need to be kind of having a look at and where there might be issues. Possibly one of the most relevant reports that we have is to directly look at the surplus and to look at the usage of all of our colonies. I do think it's important to say that efficiency across colony management helps drive down numbers, but specifically trying to drive down that avoidable and unavoidable surplus, this is quite useful. And we can look at the figures across time. We can look at different groups, or we can look at different strains. And we do this by providing clear guidance on how to clarify the fate of an animal, so that we've got good records to interrogate and then review. So here are some examples, and in that first one here, you can see there's, so pink or purple is experimental. There's no experimental use here. The pink is the breeders. So there's a lot of surplus going on here. Something's wrong. It might be that you're breeding towards a certain genotype and you're trying to find that genotype. If that's the case, you go and investigate and like, okay, that's right, but it might not be the right breeding strategy. Over here, we can highlight where there's a lot of sick animals. And so again, is that expected? Is it because it's happening after a certain age? Should we be changing the breeding strategy so that we're not having homozygous animals, or we have 2 feeder lines? And then over here, you can kind of see a variety of different experimental usage, and it's worth having a look as to the simplicity of the cross and working out whether that is an acceptable experimental use. And we can also do that by sets as well. Should look at that set data, and we're starting to share that regularly with labs, so it's available to them, but we send it out as a reminder in light of the changes to funding over the last few years, really encouraging a change in approach to where possible both sexes are used. And our scientists are required to report that both at scientific review and at ethical review. So you can see we used more females in the Crick last year than males, but that is across the whole of our research. And actually, if you look at different research areas, it varies. So another practice that drives down surplus is removing duplication of colonies. And on the wild type level, the facility provides 12 inbred, outbred, and hybrid strains that we give to the majority of the users. And we ensure that they're refreshed regularly from commercial stocks and that we try and maintain them as genetically stable as possible. We don't allow users to breed their own colonies themselves here, but we do need to ensure that we have really good communication to make sure that production meets demand. On occasion, when we do have excess, we do age stock for a few weeks, but then we normally find that they are used quite quickly because demand shifts, and we normally have a good handle of it. Where we do have excess, we often use them in our cryopreservation programs first, but we also have local animal facilities that need frozen rodents for feed that we donate them to. We also have a lot of commonly used genetically altered strains that we hold centrally. So for all of these stocks, the researchers don't pay for them. They can order them, and we normally issue them at 3 to 4 weeks, and they're provided free. Well, they're under the background cost of the facility. And we started this when the Crick formed. We review that portfolio of genetically altered strains regularly to make sure that they're still needed. We back cross and refresh them, and we have stock in the freezer as well that we provide a lot of strains from the freezer where they're used infrequently, and we'll do an IVF to provide the cross that people need. And I think providing these free is a sort of incentive to not keep their own, which makes up for the occasional mismatches in demand, of which there aren't many. We also have a tissue sharing platform where if we have an excess of animals, or if a researcher does, then we offer them out to the other researchers in the facility. I just wanted to touch briefly on the need to quality control lines, because properly characterized and maintained lines means more reliable and reproducible research, and that leads to reduction. So ideally, you need to be genotyping them with specific assays, everything in your facility when it comes in. We do a genetic background check with a mini Muga for everything that comes in, and we also work with our genetic modification team to make sure our alleles are correctly targeted. And we find several that we bring in from repositories even that aren't what we think they are. Coming to a close now, but I think it's important to highlight the use of cryopreservation and assisted reproduction as a tool to manage surplus. We provide our cryopreservation and IVF for free to help encourage a reduction in cages and costs. And you can, if you have a backup of your strain, then people are more willing to breed a lower number of animals. So if that breeding pair or trio fails, then you know you've still got stock in the freezer. Whenever there's surplus, we quite often freeze large quantities of embryos for future experimental cohorts, and that service has become very popular. We freeze a lot of sperm for lines that are occasionally required for breeding crosses, particularly Cree lines, and then we breed them via IVF. And obviously, where a line ideally isn't used for about 6 months, you would take it off the shelf. Quite often, it's a little longer than that, but and when we do have surplus embryos, we store them for media testing for plastics and oils for ourselves, but we've also offered that to others. So just a few final thoughts was providing that local guidance and signposting training, defining usage and targeting unavoidable surplus, really reviewing your colony needs regularly and sizing that colony based on the needs. Try and make it easy to comply. So providing assistance or tools helps people grow in expertise and sort of focus on the right areas, and archive your lines and communicate with your facility. So thank you very much for listening.
So thank you, Zara. I think we have a lot of questions. I do have one quick question. Have you learned anything that is really surprising from the data mining of your colony database? Something that really surprised you that you couldn't that you were not expecting, that people will use more females or more males? Or...
Oh, I see. I think definitely, I mean, I think it gave us some insights into teams that we thought were maybe not very strong, but actually we could see they were strong, but they just have very complex strains. And then where other people maybe weren't as much. I mean, I think there's lots of things that we've learned from it.
So then we quickly go to the question. So yes, is there anything special that you need to do when you deal with when you deal with aging research? How to deal with old animals? How you manage them in the colony? Or? I'm
probably not the best person to ask actually, because we don't have a lot of aged research. But we do try and make sure that we have companion animals for any of those. I mean, we do find that we have animals that have been experimented on and are then aging. And in that sense, we always try to make sure that we've got sufficient enrichment or a companion wherever possible, and that we have a higher level of monitoring.
Okay, so Isabel is asking, she's wondering whether there are some strains that are widely used, maybe at your facility. So how do you deal with them? How do you do the breeding of them? Because maybe how you keep the stock of them?
Sure. So if they're widely used, then they are managed. So we have them in a separate facility that's got a highest health status. And I mean, C57 Black 6J, for example, I've got a whole room of a couple of thousand cages just of those, I think. And I've got a couple of dedicated people on there, and they pay close attention to that stock. And we have 4 nucleus colonies that feed that stock, and we rotate them one at a time so that we maintain genetic stability, but we never have to replace the whole colony at once, and that keeps it quite stable. And then for the smaller lines that are regularly used, we make sure we're back crossing them regularly, but we try to get them to homozygosity if we can. But then we will back cross them every 5 generations, and we've got dedicated teams that really keep an eye on the demand and issuing.
So now Stephanie, I don't know whether she knows something about it. The Stephanie question disappeared. Okay, never mind. So, but Stephanie question, I think, even though it disappeared, I think it's important because she was asking whether how you make sure that all the lines are correctly labeled. I guess you check the genetics, the genotype.
Yeah. So everything, everything. So the facility is 20,000 cages occupied at the moment. We can go up to 25,000 cages. We've got about 1,500 strains at the moment on mice strains. And our database, so everything, so individual mice have ear clips. Every strain has a unique 4-letter prefix, and then every breeder has a number, every litter has a number, and every pup within that litter. So we can track every single animal. And they also have the allele information and the strain name attached to that. So they're all identified, and every single strain has its own assays, and we make sure that they are allele specific. We don't use any generic assays. Someone's asked how many breeding managers do we have? So I've only got 5 within the team. We have scientists still managing their colonies, but one of my staff would probably manage maybe 120 colonies, together with providing support to other people and building tools and guidance as well.
Okay, so maybe the last quick question, Alessandra Belgadano asking how large is the the breeding facility?
Yeah, that's 20,000 cages occupied at the moment, 1,500 strains.
Okay, thank you very much, Sarah.